A single genetic stutter, 6 letters, repeated hundreds of times, is the most common known genetic cause of both ALS and frontotemporal dementia. 2 different brain diseases, 1 mutation. This is C9ORF72. The gene is C9ORF72, chromosome 9, open reading frame 72. It sits on the short arm of chromosome 9 at band 9p21.2. Its protein helps cells break down and recycle worn-out components, a process called autophagy. But C9ORF72 is not famous for what it makes. It's famous for a 6-letter sequence buried in its first intron, and what happens when that sequence loses count. By 2011, geneticists tracing the chromosome 9 link in families with ALS and frontotemporal dementia had hit a wall. The sequence looked completely normal. Rosa Rademakers at the Mayo Clinic had spent years chasing it alongside her colleague Marielle de Jesus Hernandez. On a different continent, a team that included Alan Renton was closing in from a separate angle. Neither group knew how close the other was. They coordinated. Both published on the same day, in the same journal, in the same issue. Not a broken letter. A short passage that would not stop repeating. Both teams published on the same day, October 20th, 2011, in the same journal. Using a technique called repeat primed PCR, they found the same thing. A 6-letter sequence, GGGGCC, repeated in the first intron of C9ORF72. In healthy people, it repeats 2 to 10 times. In patients with ALS or frontotemporal dementia, it was repeating hundreds of times. Some patients carried thousands. What makes C9CRF72 uniquely cruel is that this expansion poisons the cell 3 ways at once. First, it partially silences the gene. Less C9CRF72 protein means the cell's recycling machinery runs slow. Second, the expanded repeat RNA folds into G quadruplex structures that pool in the nucleus as molecular foci, sequestering the RNA-binding proteins the cell needs to manage its own transcripts. Third, that same repetitive sequence gets read in both directions, without a conventional start signal, producing a swarm of sticky proteins called dipeptide repeats. The arginine-rich ones are especially toxic to neurons. Motor neurons die, or frontal lobe neurons die, depending on which population gives way first. Same mutation, different victims. C9ORF72 is the most common known genetic cause of ALS, roughly 40% of familial cases, and of frontotemporal dementia, roughly 25%. For families where the expansion runs, the question is not whether the stutter is there, it's when it will speak. The leading strategy has been to silence the repeat. Antisense oligonucleotides, engineered to destroy the repeat-containing RNA before it can cause damage. Early trials reached their molecular targets. The toxic transcripts fell in spinal fluid. But in patients, the disease didn't slow. The mechanism runs 3 ways, and quieting one has not been enough. Newer approaches target the toxic proteins directly or aim to restore autophagy. No treatment yet changes the course. 2011, The stutter found simultaneously by 2 teams. 2013, the toxic proteins identified. 2022, first large trials completed. 1 stutter, 3 poisons, 2 diseases. The question now is whether we can reach all 3 arms at once. The gene channel.